think it's recording. But for some reason it has this big box in the middle of my screen. I can't see around it, so I'll have to keep watching up here on that screen. Okay, so genes and genetic diseases. Kind of a fun thing, and this is not biology, introduction to biology, so I'm not going to make you memorize all of the specific details. Things you need to know, I'll rewrite down. So when you're going through the chapter, like the first 4 pages, I think, give you a review of genetics, or kind of an overview of genetics. And don't forget, this is pathophysiology, so you have to remember your anatomy, you have to remember your physiology, and then I'm just modifying it with the diseases. So I'll give you the basics of genetics again, like I said, and then we'll go over specifics. The line popped up. I put the list of objectives so that you can see what we're trying to get accomplished here. What's kind of cool about genetics is that we're transitioning to what they call the genetic age, or age of genetics. Now that we've actually decoded all of the human genetics and figured out what's in there, the major parts of it, we can go through and look and see if you have a disease, is it genetic-based? If you have certain genetic-based diseases, we may be able to actually design drugs to help you specifically, which is kind of cool because up till this point, we've really been focusing treatments like medicines on the general population. So if your genes aren't exactly perfect like the other person with the same disease, it could cause you to die, it could cause you to get really sick from taking a medication. So when we start looking at your genes and look at your genetics, we can actually make medication specifically for you so that you don't get all the nasty side effects. We can actually treat you. Some of the terms you want to remember when we talk about congenital. Congenital doesn't always necessarily mean genetic. It can be something that happens in utero. What if it's something that's potentially life-threatening? What kind of genetic situation is that? It's a malformation. Yeah. So what we typically refer to as genetic defect doesn't have to be life-threatening, but if it is, it's a malformation. Like polydactyly is too many fingers. That make you an awesome piano player and not actually threaten your life at all. All right, so when we look at the first parts, we're going to split it up a little bit to molecular genetics and actually look at the overview on how the chromosomes change too. When you look at the molecular genetics, we're looking down at the molecules of the DNA. And the DNA, remember, is made of these little nucleotides. Anybody remember the name of the 4 nucleotides? Or the letters they are? So yeah, I know, they're all there, they're just random. I always remember GCAT in that order. Good cat is how I remember it, because when they pair up, can they pair up with any of the other 3? Nope, they specifically pair G, C, and A to T. So when you look at this code, if you see a blue like this, and it's a G, you know this has to be what letter? A C. And if this green over here is a T, what does this one have to be? An A. They have to pair up like that. What's cool about genetics is that your body, universally everything we know as being alive, is made of the same 4 letters in its recipe. We have an alphabet of 26 letters that you can put a slide like this together and you understand it. But everything in your DNA, your genes, is made of just 3-letter code. 4 potential letters, sorry. And then we put the words in the 3-letter codons. We'll talk about that just briefly. So it's amazing. Every recipe is 3 letters. It would be like putting a recipe for cookies together and all you have are egg and then shug, S-U-G. And it's just little fragmented words. So we'll talk about that just briefly. And I'll give you my analogy for remembering DNA and how it works. Of course, where's DNA stored at? The nucleus. There's one other place that's kind of sneaky that you can find DNA. Anybody know where it's at? The mitochondria. Yeah, the mitochondria actually stores a little bit of DNA. It can code for 13 different chemicals that help with mitochondrial processing and energy. But the bulk of it's right here. And when this stuff bunches up, what do we call those? Chromosomes. And you can see the process. So you start with the DNA, the page up here. You start rolling it, supercoiling it, so it's called, and it starts twisting itself. You're really, really, really tight until it's to the point where in the electron microscope you can see this chromosome structure. So DNA, and you don't have to memorize this. This is just an overview again. But when you look at it, remember you have those 4 basic letters. And the 4 basic letters are nucleotides, and they're the G, the C, the A, and the T. When you have the code, the pages, the DNA itself is double-stranded compared to RNA, which is what? Single-stranded. What was the other thing that's different about RNA? What letter is different? It doesn't have a T. It has a what instead? A U. The way that always burned in my mind, so I'd remember it, is that the DNA has that special T, special letter T, and it's stuck in the nucleus. If DNA gets out of the nucleus and floats in the cytoplasm, what do you think happens to it? It just hangs out there? It's destroyed. It has to be destroyed because that means there's something wrong if it's out there and your cell actually destroys this DNA. Can RNA float around outside of the nucleus? It can. So in my mind, I always thought if it has a U, it's kind of like protecting it. The RNA has the U, and if it goes out in the cytoplasm, it's safe. If it has a T, and it goes out in the cytoplasm, you have little enzymes that will destroy that thing and shred it. The reason that, to me, this is important is that what's more dangerous in your cytoplasm, an RNA virus or a DNA virus? If you have RNA in your cytoplasm, is it going to be destroyed right away? It could sneak by with the other RNA, right? But the DNA, should there be a DNA anything out in the cytoplasm with that T? No. So the DNA viruses are typically the ones that are easier for you to kill or easier for you to fight off. Where the sneaky, nasty ones, the RNA ones, can sneak right through the cytoplasm, get into your nucleus, and merge with your DNA. So if you had to think about a virus like HIV, which one do you think it would be, DNA or RNA? It's an RNA virus, and we'll talk about that again. Actually, we may have already talked about it a little bit. We'll talk about it again now. So with the DNA structure, just remember the double helix. And it was kind of like this, a twisty ladder. And you can see the little structures up there. Now, this isn't molecular biology, so I'm not going to make you memorize the structures. But you do need to remember the four letters and DNA, G, C, A, and T. How about RNA? G, C, A, U. Oops. All right. So to put things in perspective, when you look at your DNA in one cell in your body, so if we want to look at your DNA, typically what we'll do is we'll pull a white blood cell out. We take that white blood cell while it's going through mitosis, we freeze it, and then we chip it apart, and we look at the different coiled up chromosomes. If we were to uncoil those chromosomes and look at every recipe in those recipe books, you'd actually have 1,000 page New York City telephone books in one cell of your body, in one nucleus, in fact. So that's a whole lot of letters. You've got 3 billion different letters going on there, so 3 billion different base pairs. When you see a G with a C, that's a base pair. When you see an A with a T, it's a base pair. It's like one letter at a time. And I love these putting into perspective. So it would take a person typing 60 words a minute, 8 hours a day for 50 years to actually type your DNA. And if we stretched it out, if it were the size of string, it'd actually take 25 miles and it'd be curled up into a tennis ball. So it's pretty amazing stuff. And I think this is interesting too. When you look at how much DNA you have in you, compared to a newt, we actually have very little DNA. Compared to things like a fruit fly, we have a little bit more. But an onion, holy crap, an onion actually has better and more DNA than us. Even an amoeba, super complex. Most of our DNA is crap. So they call it junk because crap didn't work really well in a science textbook. But it's just garbage DNA. It's pretty much worthless. When you look at a bacteria, most of the bacteria's DNA is actually useful, but not us. Another fun fact is that the difference between you and a chimpanzee is about 2% of your DNA. The difference between a man and a woman is about 2% of your DNA. So I'm wondering which one's closer to a chimpanzee, the man or the woman. But I've never quite figured that out. I don't know that I want to know. Okay. And then a couple important things you want to remember. So first, the DNA is the instructions for life. You get it from your parents, they got it from their parents, and it's generation to generation to generation. Does it ever change? It can change. Yep, it can change. We'll talk about that today. Changes typically aren't good. Typically. Sometimes those changes cause evolution, which can be handy or helpful. Not like X-Men evolution type of stuff where you shoot lasers from your eyes, but small things to help you adapt to the environment better. And then the second thing is that DNA contains the recipes and instructions for everything. If you don't have DNA for it, your body can't make it. Alright, so what this DNA stuff's good for? Two processes. One's called meiosis and one's called mitosis. When you look at meiosis, the only thing you want to remember with meiosis is it's for generation to generation. It's for passing on your genes. Because meiosis makes two types of cells. What are they? Or two cells. The gametes, so in a male it's the sperm and in the female it's the ova or the egg, right? So it's for passing on the genetic information. When you look at mitosis, mitosis has two purposes. Mitosis is either there to grow the organism or to fix the organism. So when you hear meiosis and mitosis, what sticks out in my mind is the I and the T. The meiosis is for individuals because when the cell replicates and splits, it makes two individual cells. If you're individual, does that mean you're exactly like your neighbor? It means you're extremely different from your neighbor. So when a man makes 250 million sperm for one ejaculation, there are 250 million different sperm. So one ejaculation could pretty much populate the United States, in case we needed it. When you look at mitosis, the T means that every daughter cell from this cell is going to be a perfect twin of the parent. That means that all the genetics should be exactly the same. They're twins. So you start with this one stem cell, it replicates into two identical or perfect twin stem cells, and then 4, and then 8, and then 16, 32, and 64. And just keep going. But every one of those cells are perfect twins that go through mitosis. So growing the organism, the other time is for repairing the organism. You get a cut on your skin, there's a big gap in your skin cells. The cells on the outside of the gap start realizing there's no neighbor touching hands with it, and then they replicate. Are those cells very different from the original cell? Hopefully they're exactly the same. Same type of skin cell, they just fill in that gap. So repairing things. Mitosis repairs where meiosis makes individuals. And then just another slide illustrating or showing it. Anyway, so I'm not going to make you go through this. This was anatomy. Through the different phases, you should always remember the different phases of mitosis. And I always remember P, mati, prophase, metaphase, anaphase, telophase, and then I. Do you remember what I was? Interphase. Of all those phases I just mentioned, the P, M, A, and the T, those are all part of mitosis. The I is its own section. I is when the cell's just alive and living and healthy. When it needs to replicate, it kicks in, starts, well, like a pregnant woman starts consuming more calories, it brings more proteins, carbs, and fats. It gets bigger, 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 and then it starts replicating. Makes sense. The big difference when you look at those phases, P, mati, is that if there's a prophase 1, a metaphase 1, anaphase 1, and a telophase 1, there must be a 2 for each of those. This is meiosis. Meiosis has all the same phases, except it doubles it. It does it twice. Because it replicates, and then those replicants split into those individual cells. Like I said, this is just an anatomy review. And then there's the cell cycle. When we get to cancer, we're actually going to spend more time on the cell cycle. But here's your interphase. Growing healthy cell, growing, growing, growing. During this phase, it starts thinking, okay, I've got to replicate myself. It starts eating or consuming more nutrients, proteins, carbs, and fats. Then, it has this checkpoint that we'll talk about can break for cancer. When it realizes, okay, I got all these basic nutrients, I'm all ready to replicate, now it starts S phase, which is synthesis of DNA. It replicates all its DNA. When it gets through replicating its DNA, it has this checkpoint that says, could we replicate the DNA? Are we sure we have the raw materials? If everything's okay, then it goes through this last phase of growth, prepping itself, and then it goes into mitosis, which is this little segment of time. This whole cycle could actually last hours to days. Growing, living, being happy, start replicating, and then maybe watch Big Bang Theory. You see the episode where Sheldon went through mitosis? Because they don't think he's capable of sexual reproduction, so he just starts eating all kinds of things, and then he starts shaking, and then whoop, going through prophase, metaphase, anaphase, telophase, and then cytokinesis is split. So, and we'll go through this. If everything's okay in the cell at this point, the cell says everything's safe, the DNA replicated, it all looks good, there's no major mutation, we have enough nutrients, we've replicated the organelles, everything's good to go, and then it starts splitting, and we have 2 perfect cells. Perfectly what? Individual or twin? Twin. In mitosis, they're twins. Alright, and then like I said, for cancer, we'll come back to the cell cycle more. When you look at the DNA replication, you have this process. And just some key players you want to remember, they can break. Number 1, you have enzymes. Enzymes speed up reactions is what you want to remember. In this situation, you have an enzyme that if I had the original copy of a recipe book, when an enzyme comes along, it opens up the pages of the recipe book, it makes a copy, and then it reseals the pages. and I keep using a recipe because that's my favorite analogy that helped get me through genetics, was when I think of DNA, I think of these recipes. My grandmother had this huge recipe book that she had, and I think it was her mother's, but it was old, it was really worn out, but it was her precious, valuable paper version of recipes. Every recipe she ever knew, she had in this. If I wanted a copy of it, did she loan me the book? Heck no. She knew better than that. What she made me do is come over, I'd bring a piece of paper, I'd open the book right in her kitchen. What would be the analogy for the kitchen where this recipe's at? The nucleus. Yep, so in the nucleus, I'd be in the kitchen, the nucleus, opening the page, and then I'd go through and I'd copy the pages that I want. And then I would close the book safely and put it back on the shelf. Because if I didn't put it back where I found it, then she'd get really ticked off. So maybe that explains a lot about me. But I would be able to go over to her house, copy the recipe, put it down, and then I'd take it back to my house. If it was a precious recipe, I'd laminate that sucker and make sure it's preserved, and we'll talk about that. And I put in my own recipe book. So when you're looking at this, you have these key players that are enzymes. They're responsible for untwisting, unzipping, opening up the recipe pages, and then making copies of them. If you ever get into genetics, there was actually this, a helicase is the thing that unzips it. And I think some being recorded out, not to tell you this joke, never mind. I think I maybe told it in another class, and that might be why you do that. But if you ever want to hear the best pickup line for a science geek, just ask me. After the recording's off, I'll tell you. Because it's funny. I dated somebody that, she goes to Harvard now. She's way smarter than I am. But she used a funny pickup line about enzymes and DNA. All right. A key term here is called semi-conservative replication. And this is a big player when you have mutations or flaws. Here's my original DNA, my original recipe book. So instead of being like grandma's where that recipe stayed in her house, what actually would happen is I would tear half of her page out and replace it with a brand new half. So the semi-conservative replication is where you open up the DNA pages, you make a perfect copy of it with brand new, and you have an old half of the page there. So that new twin cell gets this half, the original cell gets the other half. Does either of them have the original DNA completely? Nope, each of them have half brand new and half old. It's called semi-conservative replication. This is kind of good to know because when you have mutations, you've got an old strand of DNA. Could that thing possibly start getting worn out? Yeah, a cell can replicate about 50 times before it's done. This DNA gets worn out through this process, and eventually you just kill the cell. If the cell knows it's been 50 times, what process does it go through to kill itself? Apoptosis. Or apoptosis is what it looks like. But so semi-conservative replication. The next thing you want to have written down, and anytime you get in any science class that has to do with any kind of genes, this is called the central dogma of life. Whether it's insect, plant, human, any kind of creature on our planet, everything starts with DNA. Yeah, okay. So DNA. And then from that DNA, I open it up, I can either make a copy, like we just talked about through semi-conservative replication, or I can actually make a copy of the recipe to take outside the nucleus. And that copy that goes out is always called RNA. So life, everything about life starts with DNA, it turns into RNA, and every time that you code an RNA, this is the recipe that I'm laminating and taking to my house, every time it makes a protein. That's what they call the central dogma. All life revolves around these three things. DNA always turns into RNA. RNA always turns into proteins. And then proteins do everything else. If you want to make fats, the protein is the thing that makes the fat. If you want to make a carbohydrate, it's the protein that makes the carbohydrate. Heck, if you want to make another protein, you have a protein that makes that protein. It's always the same. If you mutate any of these steps or you change any of these steps, you've got problems. And we'll talk about things. Like it could be a channel to move something across the membrane, like chloride. So if you can't make that channel efficiently, that protein efficiently, it's called cystic fibrosis. We'll talk about that. I think today we'll talk about it. If you can't make insulin, what disease do you get? Diabetes. Insulin's a protein too. They're enzymes that make the insulin. Okay. So here you start with the genome. The genome's in the chromosomes, and you break the chromosomes down. One gene is just one section. So the gene is like the actual recipe itself. The chromosome is that if grandma had 3 books, this one's for making burgers. You have another chromosome that's there for making cakes. You have another chromosome that's there for making different types of salads, blah, blah, blah. And those genes, after we get that recipe, we take it and we build proteins. What are proteins made out of? They're made of amino acids, right? And they're long chains of amino acids. So you have huge proteins, you have little tiny proteins. And then the amino acids, we have 20 of them. And no, you don't have to memorize them. And when I teach Intro to Bio, the book that I use has this, and I was so excited when they had it because that was actually my But, so Grandma's cookbook is where it starts. Where's it at? Where's your cookbook at? In the nucleus. Once we get done opening this up and we actually write the recipe down, translate or transcribe? Which one means to write? Transcribe. So when you've ever heard the term scribe, to scribe something, it means to write something. So in this step, we're going to talk about this transcription, is to write that recipe down, to make a copy of it. Once that copy's made, what was this copy? DNA to RNA. The RNA slides to the nuclear pore. Once it's out here, as it's sliding out, I'm going to talk about laminating. You have to protect that thing, otherwise it can get damaged. We'll talk about laminating in here. Once it gets out here, what are these little things that make proteins? Think of your cell biology. Ribosomes. Yep, so it's going to be up with ribosomes, and the ribosomes are like the tools. You've got the mixer, you've got the spatula, you've got all the pieces that are helping you put the recipe together. The process of actually putting the recipe together, and like I said, for my grandmother, my great-grandmother, she only spoke German. So opening her cookbook did nothing for me because I didn't speak German very well. So what would I have to do to that recipe? Translate it. Exactly. So I take it from the old language. I made a perfect copy, and I took it home, and I had my dad come over and translate this thing so I could figure out what eggs and sugar were. For the translation process, I need the pieces. I need my tools, I need my recipe, and I need somebody to tell me how to put them together. That's translation. And then the final product are delicious cookies. And then, of course, in the cell, they're proteins every single time. But it's a step-by-step process going all the way through. Are all cookies exactly the same? Nope. In fact, you probably have a recipe for chocolate chip cookies. I have a different recipe for chocolate chip cookies. Genes are the same way. One recipe doesn't make everybody's cookie exactly the same. So, hair color. Some have blonde hair, some have brown hair. The genes are very similar, but they don't make the exact same outcome. And then I have each of these steps in here on the next several slides. So the first one, transcribing. What did that mean? To do what? To write it down. And these are all in your slides, but you really want to remember you've got enzymes that help write it down. That enzyme thing is called a polymerase. So what you want to remember is you've got the enzymes, and transcription is the process of writing it from DNA to RNA. Exactly. So I've put the process through here. You can see it unzips that little area. It makes a copy, and then it zips it back up. It's done. Transcription. And this is what it looks like in the cell. What's amazing, I think is amazing about the cell, is there's this little enzyme that shows the motion going through here. Relatively speaking, this thing's moving at about the speed of a jet plane. So if it were the size of a jet, it would move that fast. And it goes along reading like a little copy machine and making copies. Will this be perfect every time? That's fast, right? Do you think it'll ever have flaws? Yeah, it gets flaws sometimes. So we'll talk about what happens when flaws. For the process of transcription, you can see it recognizes the beginning of the gene, the recipe page, so it goes to page 24. It opens that page, zips through, makes a copy of it, and then shuts the page after. So it recognizes the bind, it transcribes it, and then it terminates. Once I get done with that, this is a special step. So I made the note card. What did I want to do to the note card before I actually got it home? I wanted to laminate it. This process of tapping and editing, it takes the unnecessary parts out. Like my grandmother's cookbook had doodles all over it. Do I want to copy those? No, because I had no idea what the doodles meant. You know, it might have been a 5-pack of Pall Mall smokes or anything. I could care less about that. So I can go ahead and edit those out. But it takes this long recipe and it cleans it up a little bit, makes it shorter, and then it laminates it. It puts special caps on the end of tails, so it protects it. It's a valuable recipe. I get it home and I stick it in my cookbook. That's transcription still, writing it. So when I'm cleaning out the junk, there are two terms you want to remember with the junk. So you have these things called exons and introns. The introns are junk. The exons are important. And of all the 3 billion letters, 300 billion letters that you have, only 5% of it is worth anything. Lots of junk. So if I took a long strip of that recipe book, this page is worth something, this page is squat, this page is squat, this page is worth something. Those things that are worth something are called genes. They think this stuff used to be worth something, but we've evolved and we've mutated it enough that it's worthless now. But the genes are the important thing. So the genes hold little places called exons. If I take that page, I can still trim some more of that out. Then I can just glue these two together, and this is the valuable part. What would the valuable parts be? Exons or introns? They're the exons. I always wish I had a creative way to remember that, but I don't. So there was transcription. Transcription, you open the book, you go to the right page, you write the recipe, you clean up the recipe, and now it's going to start bleeding through the nuclear pores. Next, we have to translate it. So out here floating in the cytoplasm, ribosomes are found in two places, either loose in the cytoplasm or what are they stuck to that you usually associate them with? The rough endoplasmic reticulum, which is a protein factory, making lots and lots of proteins. Translation actually requires 3 special types of RNA. You have one called a tRNA, and the tRNA, well, actually we'll talk about it, tRNA, rRNA, which is ribosomal RNA, and then the last one, mRNA. The thing we've been making so far is the mRNA, which means messenger. It's the message. So it comes out of the cytoplasm that finds these rRNAs, which make a what? The ribosome. The ribosome remembers like the machinery, the tools to make the product. The tRNA are the actual ingredients. So the rRNA is like the tools, the tRNA are the ingredients. It's an egg. It's a sugar. Flow would be flour, I guess, FOL. But these are telling you the different ingredients. So if this recipe calls for 3 eggs, I'll have 3 of these same egg tRNAs that plug in here. They're carrying the product to me. And I'll show you that process now. So translation is actually making the product. What was transcription? Writing it. Okay, so with translation, when I read that recipe, I read it 3 letters at a time. And that 3 letters at a time are codons. Codon is the same as having a word on the recipe page. And in every possible 3-letter combination represents one of the 20 amino acids. So when you look at something like proline, this one here, you don't have to know the names of the amino acids. You can see it's CCU, CCC, CCA, CCG. Any of those 4 will actually carry a proline. So if it calls for a proline in my recipe, any of these 4 letters, or 4 3-letter words will actually do it. It's kind of cool how that works. What's also kind of cool is that if I screw up one of the letters, will I get a different amino acid? I might get a totally different amino acid. That would be like having a recipe that called for salt, and you screwed up one letter and it turned into sugar. I mean, they look the same, right? Have you ever done that? Replace salt with sugar? Terrible. I actually went to one of those cooking classes with my wife, and a lady was making Irish soda bread, and she took a big scoop of salt instead of a scoop of sugar. It was terrible. I tried eating it with a smile on my face, but man, it took a lot of beer to choke that one down. Okay, so codons are like the what's? 3-letter words. They're 3-letter words that tell you what you're trying to look for. So codons like a 3-letter word. So every product has something like this. The dog ran for 1 day. There's one recipe for one sentence. So here I have the beginning, here I have the end. It's like a machine that reads 3-letter blocks all the way through. So as I'm reading this to get the ingredients off, I start with these first 3, that codes for something. Second 3 codes for something. Third 3 for something, 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 all the way through. Man, that wasn't bad right off the top of my head. It actually makes sense even. But all 3-letter words, all the way across. If you screw up this letter, what happens to that sentence? What actually happens to this sentence is the machine is going to read it. It's going to read how many letters at a time? 3. Head, auger, amp, oro, ned, and then whoops, mistake. Does that make any sense at all? It totally screwed the whole thing up. Is that going to make a good product? It's going to be completely wrong. We'll talk about that in a little bit. So these codons are really important because if you screw up one at the beginning, what happens to that whole protein? The whole protein screwed up. What about if you screwed up this letter? It would be good all the way to the end. You might get lucky and get away with it when you use that protein. Location matters. It's important. And we'll talk about mutations that happen with that kind of problem. All right, so here are your raw materials. We have the tRNA. Which RNA would be associated with this one? The rRNA. And 3 amino acids are actually stuck to the tRNA. The tRNA is like the carton that's carrying that product. So you get one carton around one egg. You get one package wrapped around 1 cup of sugar. It's the carton, the tRNA. It's carrying the product. And this is what it looks like. So here's your recipe. What's that called? The whole recipe. I'll stand here for a reason. What's this whole message called? This is a codon, but what's the whole thing called? It's the messenger RNA, right? So the mRNA is this full script. I read one word, another word, another word, another word. So if this one calls for egg, sugar, flour, but then we'll say baking soda, whatever. If this calls for baking soda, the 3-letter word for baking soda, it brings this tRNA in that fits perfectly. What always matches up with a G? A C. This is RNA. What changed? What do we not have anymore? G. So what matches up with a U? Has the A. So when you get this perfect matching, if this anti-codon is this opposite codon fits into this word, hey, you belong. And then what's going to do is it's going to steal its amino acid and dump it into the recipe, and I'll show you how that works right here. So it's just like this. Here you have your recipe, starting sequence, say start here, recipe ingredient number 1, number 2, number 3, number 4, number 5. As it reads this, it says, give me an egg. It brings that tRNA over, plugs in here, it steals the egg. Kicks out the carton, throws it in the trash. It says, give me a flour. It brings over a flour, steals the flour, throws the carton in the trash. And it keeps doing that all day long until every one of these ingredients are put in this long chain. So I have amino acid, amino acid, amino acid. What am I making? A protein. Yeah, in general, it's just a protein. I'm making this long chain of protein. And that process of making the recipe is what? Translation. Yeah. What was the process of writing it? Transcription. And I'll just put this process in here again so you can see it over and over again. I put these in because I always thought that this order kind of made more sense and it simplifies things, but I'm pointing out the main details that you have to remember. And it just shows you the steps again. If you break any of those steps of making the recipe, the whole recipe is screwed up. The cell, pretty unforgiving. You have some mistakes sometimes, but for the most part, if you screw up any part of that recipe, it's pretty unforgiving. These are the things that could happen. You get mutations. So you can either get cell death because it can't make this thing, or sometimes you get this weird mutation. And I love the fruits that are like that, like the tomatoes that have all the little tomatoes that are budding off of it. They're actually fruits, but I feel like they just belong around. So I've never seen a cherry that does that before. Okay, but a lot of times peppers. You ever done that? You open a pepper and there's like a mini pepper growing inside of it? It's a mutation. Luckily, it was compatible with life and it didn't kill it. But when you screw up that recipe, you make faulty proteins. And these proteins, a lot of times, are coding for enzymes. And if you have a screwed up enzyme, then you can't make a certain reaction work. A good example of a faulty enzyme would be somebody with lactose intolerant. What are they missing an enzyme for? They're breaking down what? Lactose is sugar. Yeah, they can't process the lactose. Anybody here know what fast flushing is or ever heard of it before? It's another fun one. I'll show you that in a couple of slides. But the Asian population, evolutionarily, they actually lack the enzymes for breaking out alcohol like Western Europeans have, or Eastern Europeans for that matter. But people from more of Europe, we can break down alcohol because we evolved that beautiful liquid ambrosia of God. Where the Asian population, culturally they're a little more pure than we are, so they don't have those enzymes. They didn't evolve with it. So when Asian people would start drinking alcohol, they get drunk really fast and they actually get really sick really fast. It's called fast flushing, and we'll talk about that. So we're missing the appropriate enzymes. Some other ones, and I think they're missing an enzyme for breaking down alcohols. So the alcohol actually sits in their body and becomes more toxic faster. Okay. Here's another one. So when you look at this is red blood cells. But the hemoglobin inside the red blood cells, you've got a chain of amino acids about 140 amino acids long. If you screw up one codon, just one, for one amino acid, this is what happens. You know what this disease is called when it gets shaped like this? Sickle cell. It's screwing up one amino acid is all it takes to get sickle cell anemia. What can't they carry very well then? Oxygen. They can't carry oxygen. The cells get all bent out of shape and they stick together. And we'll talk about that. It's a genetic disease. So this is the first part of the actual pathophysiology. We covered the genetic basics. Well, as we need a little more genetics, like Mendelian genetics, we'll talk about it in a little bit. But when you look at the mutations, these are where most of the problems happen. When you're copying one cell to a second cell, about every 1,000 base pairs, you make a mutation. You screw something up. Luckily, what's 95% of all of your DNA? Junk. It's junk anyway. So a lot of these mutations are just changing junk and it doesn't actually have any kind of effect on it. But a mutation is just an accidental error. You can get it from exposure to toxins, UV exposure, exposure to certain gases. You can get mutations from pretty much anything. When you look at the types of cells, the somatic cell is your body cells. So when you get a mutation in a somatic cell, it only affects you. It doesn't affect your offspring. What's the name of the cell that produces offspring, such as a gene? The gametes. So if you get gene mutations that affect the somatic cell, it causes things like a tumor or a cancer. If you get a mutation in the gamete, who's affected? You? Your offspring. Yeah, so if it's compatible with life, it's your offspring. And we'll talk about these different things. So sometimes you have these polymorphisms with your many changes that don't impair function. Other times you have it where it completely kills the cell. If the cell keeps living, but it's a really bad change, like it keeps replicating rapidly, what do we call it? A tumor or a cancer, right? I mentioned 1,000 base pairs. Germ cells, or gametes also. It causes mutation, and I mentioned some of these things. You can have UV exposure, you can have X-ray exposure. The early X-ray techs, they didn't know that that would screw up their genes, so the early X-ray techs would actually hold the plates. So a person would come in and just hold the plates, they'd scan them, the next person would come in and they'd hold the plate there. You'd come in and get an X-ray, you got exposed one time, but those X-ray techs would get it 50 or 100 times a day. So what were they mutating? What part of their body? Their arms, yeah, their hands and their arms, and they started getting all kinds of defects, and they'd have to replicate skin on their fingers, it wouldn't work, and then their skin started peeling off and dying. Do the same thing to their bones. So they were starting to lose digits. And they went, maybe there's something bad. Before that, a pregnant woman could actually get an X-ray to see how the baby was developing. Good idea. The types of mutations we're going to talk about, we'll talk about whole chromosome mutations or aberrations. We'll talk about single base pair changes, so changing one letter. And then we'll talk about things like frame shift mutation. And it's going to be the same thing. Like frame shift, when you change one letter, it changes the whole reading. If I substitute, what's that mean? Did I lose something completely? I replaced it. Yeah, so if I substitute, let's say that this is my substitute. Could you still make sense out of that? The dog runs for one day. You can still make some sense of it. But, like I said before, and this is a substitution. I took the letter out and I put a different letter in. Frame shift is taking one complete letter out. Is that what you're wondering? No, you lost the whole thing. So which of these two do you think are more dangerous, the substitution or the frame shift? Absolutely the frame shift. Do I not say wink wink enough anymore? Just asking. So I put the slide on here, also borrowed from the biology book, but it's on here just as a summary. So you can see what happens with substitution. You pull one out and you put another one in. Everything along here is exactly the same, just that one letter that is changed. If you have an insertion, when you pop something in, that can cause a huge difference too. Or a complete deletion. These two things fall under the term frame shift. If I add too many letters, so what if I had a stutter here? P-U-G. So, a little stutter there. Does that make sense? So it's still three letters, right? D and then dog, gra, f, ron, a day, and then that extra Y at the end. So it makes no sense. Either of those are frame shift because they shifted the whole reading frame off. If you put too much in or you take too much out, they're called frame shifts. Substitution means you pull one out, put one in. Substitute. Nice and clean. You actually have machinery that fixes your DNA for little tiny problems. So if something little like this were to happen, you have a repair mechanism that can fix that. So, ooh, that's not right. Let's shuffle this and try one of those other letters, and it can scramble a little.
So what would happen in the substitution of just that one word with the substitution?
It could make, you could get lucky. You remember how when we looked at proline, there are 4 different combinations. You could get lucky, and you could still make proline, and that mutation didn't change a thing. You could get a little bit unlucky, maybe it changes like proline to a leucine, a different one, but it still works somehow. So they're similar enough that it still works, or it can change to something completely wrong and screw it all up. Yeah, so there are really 2 major problems. Either you accidentally get lucky and you make the same thing, or you just screw the whole thing up. Yeah, but frame shift, rarely forgetting at all. Because if you change all of that stuff, it screws everything up. Our genes, we need repair mechanisms, but these repair mechanisms aren't all that good. And when you look at other creatures like sharks and great apes, they haven't evolved in hundreds of thousands of years. They're the same creatures they were 200,000 years ago. With us, we've been evolving gradually forever. So it's slowly changing, I guess, depending on your perspective of life. But we've been slowly changing because human DNA sucks. It's weak. Our repair mechanisms are terrible. Is that always a bad thing? It increases our risk of cancer because it causes lots of mutations, but it also helps us adapt to our environment. So bacteria is the same way. Bacteria, their DNA kind of sucks. I mean, they've got a lot of DNA in there, but their DNA is so flawed, they get a lot of mutations. But they can evolve and change really quick, too. So what's that tell you about things like great apes compared to humans? What's happening to apes compared to humans? They're becoming extinct. Yeah, they're not evolving. They're not changing their environment. Their environment around them is changing constantly, but they're not changing at all because their DNA is too good. Ha, sucks to be you. Oh, I forgot. We had the last one. And then here with the chromosome aberration, this is where you lose whole sections of chromosomes. So you can change the gene. You delete the gene completely. If it's gone, can you make that thing anymore? Nope. So let's hope you didn't need it. Okay. So you can relocate a gene. You can actually take a gene from one part of the chromosome and plug it into another. And there are some good or bad things that happen there. It's too bad, but so we'll talk about that. Sometimes you can accidentally make 2 of the same gene. So when you start making a copy of this, you make 2 of it. It's like having 2 batches of cookies every time you need a batch of cookies. So could that possibly be bad? It could, yeah. It could make you overproduce some things. If you build up certain things that you don't want built up. And we'll talk about that too. So this is kind of like an overview of the problems we're going to talk about. And then this was just another slide that showed examples of frame shift. But this one's from your textbook. I like the other one better. Anyway, if you knock a whole letter out, you can see the whole thing is shifted off and it changed everything in that reading frame. Okay, so there are 4 basic steps in the mutation. So from normal to illness, and just 4 easy steps. First, mutate a gene code for a non-functioning protein. You screw up a letter, you screw up the recipe. You replace salt with sugar. That's the first step. You screw up the DNA or the RNA. The second one, since you don't have that enzyme, you can't create the reaction you originally wanted to. So we'll say that in Asian culture, somewhere along the line back in the day, they had a goof up to the enzyme that processes alcohol. I'm sure there was some evolutionary benefit to that. Like the ones that could process alcohol, they became alcoholics, drunk, and then Darwin took them out. Well, Darwin was took them out. And those are smart ones, right? So they evolved well. And ones that were more pure survived. But anyway, you can't process that thing. Next, the molecule, the enzyme, would normally process starts accumulating. So if this were breaking down alcohol, what's going to happen when they were exposed to alcohol? Yeah, and by the way, there's alcohol in other things other than just beer and vodka, right? So you can't process things that contain alcohol very well. And the fourth step, that chemical when built up can actually kill them. What's kind of fun, seems like a weird word to put in this, but what's kind of interesting is that there are enzymes, like that alcohol breakdown enzyme, that they have specificity. They're proteins. They're specific for certain things. They're like a lock and key mechanism. If they're specific for alcohol, they fit things that are alcohol or like alcohol. Here's a fun fact so that you have a story to tell your friends. If you spill, what's the stuff that they put in your radiator? Antifreeze. Over my head. But anyway, if you spill antifreeze on the ground, you ever seen dogs and cats around antifreeze? They love it. It tastes sweet. It's like a sugar-based alcohol product. The bad thing is that it's very toxic. So they see this, they smell it, and they start drinking it because it's sweet and it's tasty. It goes to these enzymes that process alcohol, it sticks to them, and it clumps it up and clogs it up in their liver and kills them. So they die. What's interesting is that if you take your dog to the vet, what could they give them to bind to that enzyme before the antifreeze does? An IV of vodka. Yeah, if you go to the vet clinic, I was at one of the vet clinics, the emergency clinic, and they actually keep a bottle of Everclear. And they IV Everclear into the dogs until they're smashed. Like, so smashed they can't stand up. They're just out of it. But the good thing is that if the alcohol binds these enzymes first, it's safe. And then the antifreeze gets peed out, and then the alcohol eventually breaks down, and then the dogs have a hell of a hangover the next day.
Does the antifreeze and alcohol have equal chance of binding, or does
They do. Yeah, they have so their affinity is very similar. That's what you're asking, right?
Yeah.
Yeah. And so what they're trying to do is they're trying to outnumber. Yeah, get them more alcohol than they have antifreeze in them, so it wins. Yep. And then this is an example of the fast flushing. Huh? Yeah, it would if you drank antifreeze. So I'm thinking why somebody would, but you know what? There's always somebody out there. Like, do you remember a couple years ago? Yeah, there was an episode of The House about that where the guy drank ink toner, and then House got him drunk. It was LL Cool J was the actor. House got him drunk, thinking they were just last drinks, and he actually cured him with alcohol. But fast flush. Oh, I was thinking, man, that would be an excuse if a kid drank out. It shouldn't be reported, but if a kid drank alcohol and said, well, I drank antifreeze, they'd have to drink alcohol, right? They get smashed. Really sick. Get smashed.
But with alcoholics, aren't they also missing some sort of enzyme that gives them prone to alcohol?
There you're touching it. It's not an enzyme that makes it prone to it. They think there's hereditary features like in the brain. But no, after you drink alcohol, you start making more and more of this enzyme so you can handle more and more, which builds tolerance to the alcohol. That's what you're thinking. But in this situation, she has one drink and then you can see her face turns to flush because she can't process the alcohol. So they get this fast flushing syndrome. Their blood vessels start dilating and opening up because of the toxins in their body. It's an interesting thing. Okay, so gene mutations. I already talked about it a little bit. You can have a nucleotide change, which is one layer that's off. You can actually have, if you change that one nucleotide in one gene, they call it a single gene disorder, because guess how many genes it affects? A single gene. Yep. You can have polygenetic, which affects lots of them. We'll talk about things like that with people like cleft palate. But single gene disorder, these are nice when you're taking a genetics test because they're easy and very straightforward. It's one gene, one flaw that's in it. So they're called single gene mutations. Here's an example. Tay-Sachs. This is a terrifying disease, and this one you'll want to know. I think I mentioned it the first week too, but with Tay-Sachs, it's a genetic disease, so how did the kid get it? Passed down from mom and dad. Occasionally there's a mutation that might accidentally cause it, but typically it's passed down from mom and dad. And a lot of these single gene mutations, you find them very commonly in like certain ethnic backgrounds or certain cultural backgrounds, like Tay-Sachs, Ashkenazi Jews, because based on the religion, historically, they only married other Ashkenazi Jews. So if this disease was in that community and they kept mating with people that had were carriers for this, there could be more and more recurrences, Tay-Sachs. The Amish are like this with polydactyly, having an extra digit. So it's a gene mutation, they get an extra digit. It's more common in the Amish community than the general population. So a lot of these single gene disorders you actually see in a lot if it's a community that marries into their own quote-unquote people. So whether it's race or ethnicity or religious background, you see a lot of it. But Tay-Sachs is one of those. And I remember watching a video for this a long time ago about this couple. They had a child that had Tay-Sachs. And they didn't know anything about it. And they started looking into their background, and they found that both families carried it. So they had another child. That child had Tay-Sachs too. And then the guy's brother had a child, and that one had Tay-Sachs. Man, that was intense. It was sad. Jim?
What about sickle cell anemia? Is that also single gene?
Yep.
Okay.
Yep, and we'll talk about that one too. So, yeah, actually, I'll talk about that then. But with Tay-Sachs, in that situation, what happened is that their child was born, and this is terrifying because the child starts developing until about 2 years of age. They're growing, they're learning to feed themselves, they're learning to walk and talk, and they're doing really well. But what's happening is they're missing an enzyme that processes lipids. So lipids get inside their cells, and they start fattening the cells up and making them bulge. And the primary cells that it affects are neurons. So as neurons fill with lipid, what is lipid? Is it a conductor or an insulator? It's an insulator. What's the purpose of a neuron? Carry electrical signals. So as you're filling the neuron up, what's it going to do with its signals? Slower and slower. So around the age of 2, you start seeing their neural progression regress. So they stop walking like they used to. They start going back to crawling, and then eventually they can't crawl. They stop feeding themselves, and they have to be fed, and eventually they can't feed themselves at all. And what's happening is for about the first few years they're growing, and then suddenly it's like they stop growing and they start going in reverse. And by the time they're 5, almost all these children are dead. So it's really sad. But this family, since both the dad and the mom carried it, roughly 50% of the children don't have this disease, and we'll talk about that. But Tay-Sachs is just one of those examples of single gene disorders. Okay. Breast cancer is another one of those. So we've isolated a lot of these genes. So the BRCA gene, we'll talk about this more in the cancer section, but it's one gene that we look at, and that if they have this one flawed gene, it increases the risk for cancer. Does that mean they're absolutely positively going to get the cancer? No. So sometimes it doesn't mean it's an absolute death sentence. Sometimes it just means they're more prone to it. So they might want to change some of those risk factors that they have. Like, can you change aging? Race? Sex? Genetically, no. So you can't really change those different things, but you can change environmental factors like smoking or the toxins you eat and stuff like that. And we'll talk about this next week, but it's from a single gene being affected. And we'll talk about some of those different changes in the sequence, but these are just a couple ideas of single gene disorders. When you see mutations, when you look at mutations, we're going to talk about the chromosome and different spots in the chromosome. We'll go to the whole time today. You can look at the different spots, and what's kind of interesting is since we've done genetic coding, we know the different parts of the chromosome for different types of diseases. So we know where hemophilia B is at, Christmas disease. We know what's going on with Fragile X. We know where a lot of these different characteristics of disease are flawed and coming from. So we can actually take some of those out, do an analysis, and see if that child can have the problem. The problem with the genetic part is that sometimes you get spontaneous mutations, which means that the parents didn't pass it down, that something just went wrong. They could have been exposed to X-ray radiation. They could have been exposed to some kind of a toxin like cigarette smoke or alcohol while they're developing in utero. Sometimes they're just parts of the gene that are flawed. We call them mutational hotspots. The gene's not very strong there, so we get a lot of change. So spontaneous means it just spontaneously happens. It wasn't because it was passed down. And then hotspots are those kind of weak areas that we want to think about. Like when we talk about Fragile X syndrome, it's telling you right in the name. It's a fragile spot. It's a fragile part of the chromosome. It's prone to mutations. And a lot of times in these mutational hotspots, you can start accumulating more and more and more mutations. We'll talk about it in Fragile X then. So mutational and spontaneous. Actually, this is a really good spot to stop, because then we'll start talking about karyotypes on this next slide.